When we think of human-induced environmental change, we oftentimes focus on a single mode of change, such as rising temperature, light pollution, exposure to contaminants, and etc. While these individual alterations may be potentially destructive, in most cases, habitats experience multiple changes at the same time. And when these different environmental changes interact with one another, the way they influence animals may be more complex than when these changes occur individually. In our study, we examined if the combined presence of poor visual conditions and plastic pollutants can affect animals' ability to respond to visual cues. Due to human impact, many aquatic habitats experience increasing levels of turbidity and eutrophication, which eventually result in poor visual conditions. With our previous research, we know that the extended exposure to low visibility resulted by turbidity or dim light can impair vision, and also it can alter which sensory cues that animals rely on. For example, fish tend to respond more to smell of food than to the physical appearance of food when they live in poor visual environments. Plastic pollution is another major concern related to human impact, and because of the widespread use of plastic products, many aquatic habitats experience disturbing levels of BPA, which is commonly used to synthesize plastic. BPA mimics the function of hormones and is known to affect animals via various pathways, such as learning abilities, metabolism, and reproduction. But we know very little about whether it has any impact on sensory systems such as vision. We use zebrafish to test how the combined presence of poor visual conditions and BPA affects animals' vision. Zebrafish is a small fish that is widely used in biomedical research, and they naturally occur in parts of Indian subcontinent across a wide range of habitats such as lakes, rivers, and irrigation canals. Due to human activities, these habitats often experience turbid conditions and plastic pollution. And these pictures that we've captured during our field visits to India show the murkiness of zebrafish habitats and how common the plastic pollution is. In our experiment, we kept zebrafish larvae in low light conditions and low concentrations of BPA for 7 days since fertilization. We selected low levels of light and BPA manipulations, and the BPA concentration that we used in this study is actually considered to be environmentally safe. We tested zebrafish's ability to respond to visual cues by presenting them with moving black and white bars. And if they see these moving black and white bars, they swim parallel to it. Interestingly, we found that zebrafish that were exposed to both low light and BPA had the weakest response to this visual cue. But when they were kept only in low light or BPA treatments, their response was not affected from the baseline response. What we see here is that when multiple environmental alterations occur together, even at low levels, they are capable of affecting animals. With the presence of BPA, even a small decline in visibility was enough to impair zebrafish vision. This finding highlights the importance of considering the impact of multiple interacting environmental stresses when we try to understand how animals will survive human-induced environmental change.